my channel Pure Biology, and today's topic is the photomorphogenesis, not the process of the photomorphogenesis, which factors generally involved in this photomorphogenesis. Today's topic is that. This is the phototropin. Before going to this topic, phototropin, first we have to know what is the photomorphogenesis. What is called the photomorphogenesis? Actually, we all know the plant's activity, that is photosynthesis. This is related to the light, or you can say that photosynthesis is dependent on the light. So, not only photosynthesis, different growth and development or activity of the plant's body that are obviously related to the light. Generally the blue light or the red light, or you can say the blue and red color of the light. Okay, so phototropin is the blue light factor. That means it is pigment related to the blue light. That means its activity that controls the plant's development and the growth totally dependent on the blue light. Clear? So here first, this is called the photomorphogenesis. This is the photomorphogenesis. Generally, that means the growth and development when it is light related, or the plant's body's activity, some activities that are dependent on light, then we can say this term the photomorphogenesis. Generally, three factors very important, or three pigments you can say. One is the cryptochrome, phytochrome, and the phototropin. Okay, three are very important factors. So before this video, I already explained the cryptochrome, and here I only explained about the phototropin. The phototropin, actually, this is the activity or mode of activity, you can say. The absence of light, this is the situation, and the presence of light, this is the situation. So you can say the love 1 and the love 2. That means LOV1 and LOV2. These are the two domains. Okay, LOV1 and LOV2, the two domains that are very important for the phototropin. So what is saying here? This mediate phototropism and chloroplast movement. First, here I just saying that brief functions of phototropin, you can say. It helps in the chloroplast movement. What is the chloroplast movement? Actually, you all know that chloroplast of the plant's body, it can absorb a particular wavelength light, and if this wavelength or more than this wavelength light is absorbed by the chloroplast, then the damages is seen. Obviously, damages already occurred in the chloroplast. Okay, so here this is important role of the phototropin that control the shifting of the chloroplast according to the light intensity. Okay, according to the light intensity. Next, two kinds of phototropin. Generally, we can see one is the PHOT1 and PHOT2. Okay, so here the two domain you can see, one is the LOV1 and LOV2, and this is the N terminal region and this is the C terminal region, and this region is known as the serine. This is N terminal threonine kinase, serine threonine kinase. Okay, this is the part of the subunit, you can say, serine threonine kinase, and it is attached that between the C terminal region and the LAV2 domain. Okay, this is attached here. The N terminal contains the two similar domain. Okay, so the LAV1, that is LOV1 and LOV2, two domains generally are attached to the N terminal region. Okay, generally attached to the N terminal region. Next, what is saying that LOV, what is the full form of the LOV? LOV is known as the light, oxygen or voltage. Light, oxygen or voltage, and each contains generally 100 amino acids. That means LOV1 that contains 100 amino acids, and LOV2 that contains also 100 amino acids. Okay. So what happened during the activation? These domains bind to the flavins and have the sequence similarity to protein involved in signaling and in bacteria and the mammals. Okay, so this is seen generally, this phototropin, this pigment is seen bacteria and also some mammals, obviously. So what happened? These LOV domains actually that bind to the flavins, okay, this bind to the flavins and the sequence remain similar. What type of protein? This protein that present in the bacteria or where this phototropin present like the mammals. Okay, that help in transferring or in transferring, you can say that helps in the transferring of the signaling. Okay, or you can say that it helps in signaling. Okay, transduction of the signaling. The N-terminal half of phototropin that binds the flavin mononucleotide. So what is saying that the half of the phototropin that is bind to the FMN. Okay, FMN. This is called the flavin mononucleotide and undergoes a blue light dependent autophosphorylation reaction. So you can see here that this domain it is bound to the FMN and it is bound to also the FMN, and here main role that is found in the binding of FMN. Okay, so this situation generally arises when the light is absent, and this situation generally arises when the light is present. Okay, so when the light is present, what is saying here? The domain 1, LOV1 that form a bond with the FMN and LOV2 that form also bond to the FMN, and this forming of bonding or this bonding formation helps in the activation of the phototropin, or you can say the autophosphorylation is happening. Okay, autophosphorylation is happening. And generally in the dark situation, FMN molecule is non-covalently bound to each LOV domain. This is non-covalent bond is not seen here. Non-covalent bond is not seen here. You can see that. But in the blue light, in presence of light, or you can say that blue light, what happens? This FMN and LOV1 and FMN LOV2 both form the covalent bond between them. Okay, both form the covalent bonds. And so what happened after that? To cysteine residue to phototropin molecule and forming the cysteine flavin covalent abduct molecules. Okay, abduct molecules. This is complex, not molecule, complex. So what is saying here? Here the bonding is formed. You can see that bonding is formed, and also this bonding, that is this complex also that bound to the cysteine residue. Okay, and when this totally that bound to the cysteine residue, then a complex is formed, is called the cysteine flavin covalent abduct residue or complex. Okay, so what is saying here? That in the absence of light, this general binding we can see here. Okay, no changes is occurring. But in presence of light, here the covalent is bond formed. You can see this one and this one and this one and this one, between them the covalent bond is found. And also the cysteine residue that is also bound to that cysteine residue. And actually the complex form is called the cysteine flavin covalent abduct complex. And this formation, conformational change creates. Okay, conformational change create in the phototropin. And what happens for the conformational changes creating? That triggers the autophosphorylation, a sensory transduction cascade starts. That means When the complex is formed, in that situation, the photophosphorylation, that is autophosphorylation, it is happening in the phototropin. Okay, and as the autophosphorylation reaction occurring in the phototropin, then the signaling transduction cascade that starts, or sensory, you can say, sensory transduction cascade starts. And as usually, the activity of the phototropin that will be increased, and then at that time the phototropin that control the different activity of the plant's body, just like the movement rapidly, chloroplast movement or chloroplast shifting, and second one, inhibition of the growth of the etiolated seedlings. Second one, third one, the leaf expansion. These activities or these functions all done by this phototropin when it becomes autophosphorylated. Okay. So here we just learned here the how the chloroplast shifting is continuing or it is done by the phototropin. It is today's topic not included in here, but I also tell you or explain how this chloroplast shifting is actually occurring. Okay. So under the weak illumination, the chloroplast generally, weak illumination means when the light is very weak. In that situation, the chloroplasts that gather at the upper and the lower surfaces of the mesophyll cells. But in case of strong illumination, when the light is very strong, then the chloroplasts move to the cell surfaces that are parallel to the incident light, and in this way minimizing the light absorption and avoiding the photo damage. And these two things is totally done by the phototropin. Okay, phototropin pigment help in these two types of situation occurring in the chloroplast. Okay, so total this is done by the phototropin pigment. So today's topic is the phototropin pigment. Now it is ended here.